Welcome to the Harpe Bioherbicide podcast. I'm Dr. Alex Danis, and for today's episode, we'll be speaking with Harpe's CTO and Director, Dr. Chad Brommer. Chad is an award-winning scientist with extensive background in crop science and plant physiology, and holds multiple patents in the areas of agriculture and pharmacology. He's going to tell us about the science behind Harpe Bioherbicide, and what really goes into the development of novel natural herbicides that can support today's agriculture. Well, thank you so much for joining me to chat a little bit about Harpe today. First things first, can you introduce yourself, tell me who you are and what you do?
My name is Chad Brommer. I'm the Chief Technology Officer for Harpe Bioherbicide Solutions.
Fantastic. What is Harpe?
Harpe starts as an idea or a concept of using natural products, plant extracts, and the molecules that are in it in order to control weeds. Weeds in the soil or seeds, as well as weeds that may be growing, as we call it post-emergent. So Harpe Bioherbicide Solutions is a company that's put together to develop an entire platform of these natural products, be it extracts or molecules, in order to help in the constant battle for weed control.
And that sounds like a true constant battle. You know, farmers are dealing with this every day. Can you talk a little bit about the challenges that farmers are facing when it comes to herbicides?
Yeah, with the Roundup Ready revolution that happened in the late 90s was great. People had a new tool to spray over the top of their soybeans, their corn, their cotton, along with lots of other new molecules that the companies came up with. So weeds were being controlled, it was cost effective, it was time effective, and then over about 10 or 15 years, different types of herbicide resistance continue to develop. And that continued development of resistance means it's even harder. It takes more time for the farmers to control their weeds, it takes more planning, oftentimes costs more, can be definitely yield debilitating if they do have a weed escape. So over each successive year or 2 years, the problem in the U.S., the problem globally just gets a little bit worse.
So you are coming at this as a scientist. How do you look at that problem and say, we need to do something different, we need to do something innovative, we need to take an approach nobody's taken before? How did you go from sort of seeing that problem to creating Harpe?
Yeah, it's any time that you think about problems, whether it's in agriculture, environmental science, biology, medicine, anywhere, I think the important first step was identifying that there was a problem. And it's not just that there was a problem, but it didn't feel from an industry side that it was being truly identified exactly what is our problem, and then effective solutions were also not being developed. Now, when I say effective, I mean not just the fact that something is going to work in order to remove the weeds, but something that can be utilized in a shorter time frame, say a few years rather than 15 years that it can take for a new molecule, or into the other types of mechanical or other tools that are being developed that are highly effective, but are they deployable? Can they be used across several 100 million acres of crops in the U.S. and Canada, as well as smaller maybe acreages in some places in Europe? And so, what is the problem? What are some ways that the problem can be addressed? And when you start there, then it's just a question of what are the other criteria. Finding something that's highly effective to control weeds, but also something that can be registered, something that from an environmental side, from a sustainability side, is going to fit with the demands that the public has for their food supply, and again also to those growers, something that's going to be cost effective, something they can use in their current equipment. They don't have to buy new things. So putting a lot of different challenges on our own team to find some different solutions and pathways that are different. And so that's where some of the different plant extracts come in, easier to register, easier to deploy than maybe some other types of solutions in the biologics world. Making sure that we find something that's efficacious, and our products work in the soil as well as on the plants, and then continuing to develop those products to make them more effective, more cost effective, more deployable. Because if you have a solution that's fantastic for one place, is good. But if it's not really deployable across geographies, then it's only really a solution for one subset of people. And again, that's a grand challenge to think about finding a solution that could be deployable globally, not just within a continent or a country. You start with those ideas, and then you slowly but surely whittle your way through potential options and more options, and then you find some that are going to work, and then you continue to develop those. And that's the path that we took. So there's nothing magical. There's no, there are no light bulb moments in this as much as these are the steps that you take to make something or to build something. And as long as you know what the problem is and you're very clear about, where your goals are, you can put that together.
You say that there's nothing magical or no light bulb moments, and I get that, because it's not magic, it's science. But you had the entire possibility of all the plant extracts in the world to look at. How do you then sort of focus in on a couple or one or two that are really great, and sort of bring me back to those moments when you were testing those out and figuring out, like, oh, these might work, these might actually be the ones?
Yeah, it goes back to the criteria, because there are lots of different ones. I mean, there's different organizations globally that have been looking at natural products, plant extracts for 50 years. I mean, if you really go back, 100 years or more ago, before there was a lot of chemical crop protection agents, people were looking for natural things that farmers would use in their environments to help get rid of pests or keep weeds away and different things of that nature. So it's not that a lot of those pieces aren't out there. Again, it goes back to the criteria. And so some of the criteria, again, goes back to the deployability. And there are actually some good examples of, if there's a plant that's growing, let's say, in a remote island in the mid-Pacific Ocean,
sure,
that's great. If you find the molecule, that's great. You could probably have to hope that you can synthesize the molecule, because based on how the plant grows within that environment, it may be quite difficult to get it naturally.
Right.
Maybe you have to do a fermentation process. So each one of these things magnifies the ability to deploy, the ability to take an idea that's been proven with a molecule, with an extract, and bring it and put it in someone's hands. So we needed to make sure that at least the core set of the 10 or so extracts that we use was accessible. And in the early stages, just so we could purchase them.
Sure.
Happy to go to the Pacific to get something from an island, but that, in order to do some early stage testing, you need to be able to buy things.
Yeah.
Knowing that there was a commercial supply of a lot of the extracts that we were looking at meant that when it comes to formulating, when it comes to really looking at a deployment geographically for a certain type of crop or a certain type of application, that supply chain could be met. Otherwise, again, from a commodity side, it's very expensive going to the Pacific Island to get the one plant extract, and it's also restrictive.
Right. Can you tell me a little bit about sort of some of the main plants that you're using in this? I believe they're all sort of mentha in the genus. Is that the genus or the species? Correct me if I'm wrong. Is that the genus mentha?
Yeah.
Okay.
Yes. So a whole series of different plants within the mentha genus, different species that we've looked at that all have unique sets of molecules, some the same, some different, that can be deployed or utilized in different ways to help control weeds, whether it be in the soil or from a post-emergent side. Also look at a series of different plants that may produce somewhat similar molecules in the same chemical family. So if you're a chemist and you went to a lab and you found some activity in a molecule, you may synthesize a whole series of other similar molecules. Well, that's not really what we do.
Right.
We're looking for what's available to us from a natural side, from nature. So we sort of go to nature's cabinet, knowing that some of these plants, through their secondary metabolism, are going to make a lot of these molecules that are similar, and just start to sort of collect those, do some testing, and see what's going to be effective or not effective.
And can you talk a little bit about that collection process? Because you described it to me earlier as plant juice. So tell me a little bit about the plant juice. How are you getting it? How are you making it? And then what does it do when you spray it on plants?
Well, juice in the sense of, the plants, the extracts that we use tend to be mostly from steam.
Right.
So it's high temperature extract, so it's not sort of chemically extracted. There are different molecules and things you could get doing those other processes, but that's not what we focus on. So once those things are extracted, they're also analyzed, so we have an idea of exactly what molecules are in there. From that point forward, then we start to build out what would we use to spray on the soil, on another plant, doing the early stage testing to see what is going to work, what's not going to work, what are ways that we can make it work better. And from there, we also do a much deeper dive into how is it working. And that's where we're not really binding to a specific site of action. It's not an enzymatic site or a physiological pathway that we're disrupting, so much as that we're causing a lot of fluidity in the membranes.
Okay.
And membrane fluidity is not breaking the membrane, but it means that the membranes can leak. And so something like a chloroplast that's leaking can cause a lot of problems in the plant, similar to some of the herbicides that are used that target the chloroplast, or fluidity in the mitochondria, fluidity in some of the other parts of the plant cell are going to cause the plant cell not to function terribly well.
So we're using this new herbicide, or Harpe is developing this new herbicide that is creating this fluidity in the membrane. It's targeting sort of these multiple sites of action in the plant. How does that translate to resistance? Does it make it harder for the plant to become resistant to this herbicide? What does that mean for the farmers who are going to be spraying it?
Well, having a new set of sites of action and different types of modes of action is going to be very helpful. We're not aware of any weed that has resistance or any plant that has resistance developed to the products. That doesn't mean some plants aren't less sensitive to the products.
Sure.
So what it does for us in putting a product together is we know that if we spray something by itself, just with our product alone, that we should get very good activity. And we do across plant species, across size of plants. Even with herbicide-resistant weeds, we haven't found any to this point that we can't control. The true challenge is being able to pair our products with other herbicides, and thereby, hopefully, and we've seen this in some of our trials already, disrupt the resistance process. So by having multiple effective sites of action, we can hold off nature from developing resistance in the certain weed species. So it's not just that we have effective products or new sites of actions, new modes of action, but also that we have a very strong sustainability and stewardship message going forward about pairing products and herbicides together.
Can you talk a little bit more about that in sort of the message of this? What do you think that this kind of innovation means for the industry at large? Is this going to spur other people to sort of think in new ways? Is it going to sort of encourage more people to be thinking about sustainability? Where do you sort of see this fitting into that broader picture?
Well, within the broad sense of developing something that's not chemistry or not traditional chemistry, I think it is a general challenge, but not a challenge in challenging the way that companies are currently developing and finding products. It becomes another pathway for them to use their vast science resources and the incredibly intelligent people and teams that they have to maybe build a new pathway.
Right.
And we're already seeing this not quite as much in weed control, but with pathogen control and insect control, that companies are building out biological discovery platforms. And so the revolution, if you will, in not just using chemistry, but using some natural things has started. I think, our hope is that we'll continue to push companies and push the envelope for them to develop even more products, more sustainable products, new sites, new modes of action.
I love that. I love that this is not just one specific, portfolio, but rather sort of a new way of tackling this problem and hopefully inspiring people to do that. What are you most excited about when it comes to the science around Harpe? What is the thing that makes you go, yeah, this is cool, and I'm so excited that I'm working on this?
Well, we're happy that it works.
Sure. That's a big one. That's a big one.
Oh, I think it goes back to, a different part of the question that you ask in terms of how does it change the way that people look at things. Well, it's very easy as scientists, we get trained in a certain way with certain faculty and certain programs at certain universities or colleges. We kind of default into those processes. It's, sort of scientific muscle memory. So things like this, I think, are a good challenge because it forces people to be critical about what we're doing and ask hard questions. But it also forces them to rethink how they're approaching answering their questions and finding solutions to their problems. Now, we're a small company. We're not a multinational, crop protection company. So the advances and the things that we have done, if you start to think about what would it mean if a company that spends $1 million or more a day on research and development Where does it go from there? So to me, that's the kind of the exciting part, is that we've only scratched the surface, and we can't possibly deploy the resources that, you know, a large crop protection company could. But if they did, then it becomes really exciting.
Let's say I drop $2 million a day of research money into your lab right now. What's the thing that you would run at that you would be so excited to be like, this is the next phase for Harpe. This is where we're going. This is what we're going to spend 100 people a day working on.
Oh, that would probably be a secret. But
okay, all right, that's fair. That's fair.
No, but to that point, you know, the ability to screen even more extracts, to look at more possible herbicide combinations, so we can tailor for just in a really good example of the sugar beet example. There's not a lot of products you can use in sugar beets. It's a very important crop, particularly like in the upper Midwest and Canada. We could specifically tailor a product for sugar beets.
Right.
That would be fun. The other side of that is probably even more research into the sites and mechanisms of action, giving it a deeper biochemical, molecular biology dive.
As a molecular biologist myself, I approve of that message. I like that. That's what I would want to hear.
It's really important to realize that discoveries can be made by individuals, but products and things that are used in the marketplace are created by teams and companies, people that have expertise in lots of different areas. And that's where we've had great benefit, is we've got a great team of people to help do the research, do the agronomy, but also the business side, and understand from formulations to supply chain how all this works.
Well, I love that message of team being the one that we end on. Thank you so much for chatting with me. This was lovely. Mm-hmm. That wraps up this episode of the Harpe Bioherbicide podcast. Special thanks go out to Dr. Chad Brommer and, of course, to our listeners. For more information on today's topic and Harpe Bioherbicide's mission to provide a new environmentally safe and sustainable approach to weed control, visit harpebio.com. I'm Dr. Alex Danis, and we'll see you next time.